Thank you, Patricia. That was a very kind introduction. It's indeed a pleasure to be here, and thank you very much for asking me to give this talk. Let me share my screen. I'll try and speak slowly because I understand that interpreters are working behind the screen. So let me first share my screen. All right. So the title of my talk is slightly long. I'm going to talk to you about some of the commitments and activities of the Human Cell Atlas that's meant to promote equity, which is something very important. And an equity working group was formed early on in the HCA. And I'm also going to share with you some results of our work, single cell transcriptomics work on a specific disease, which is oral cancer. So one of the clearest messages from genomic studies is that most diseases, or at least many diseases, are much more heterogeneous at the cellular and molecular levels than was originally recognized. And this heterogeneity impacts on clinical outcome of these diseases, that in turn impacts on management of these diseases, response to treatment, and survival of individuals who have these diseases. So heterogeneity impacts on various kinds of outcomes and impacts on management of diseases. We therefore must understand heterogeneity at the molecular level. At this time, for most diseases, uniform therapy is prescribed. That is, the same treatment is prescribed for all patients. And the goal is to be able to risk stratify based on molecular understanding of these various kinds of diseases, to be able to risk stratify the screening and eventually the treatment for these diseases. There will be, if these are risk stratified, there will be reduction of overdiagnosis of these diseases. And then if the treatment is tied to the genomes, then there'll be more personalized interventions for these diseases. All of this, we believe, will save expenditure on treatment and overall increase in the number of years of disease-free survival. So essentially, that's what the Human Cell Atlas is intending to do. But to be able to understand diseases, one needs to understand normal health, because after all, normal health is perturbed in precipitation of these various kinds of diseases, and therefore initial focus or initial emphasis is essentially to understand the normal human body. Precision genomics is what we've been talking about, understanding diseases at the molecular level, tailor-making these kinds of understanding to treatment, therapy, etc. Now, has that actually penetrated into the society? And is it penetrated and is it significant in some ways to promote health equity? Right now, as we know that, health management, health dissemination is not very equitable. And what we believe is that if there is penetration of precision genomics, it will promote health equity. This penetration has not happened yet because the understanding is happening just now, and therefore the penetration will take some time, but the penetration has begun. In terms of precision genomics, currently the pinnacle of precision genomics is the Human Cell Atlas, which essentially tries to understand or define human body cell by cell. Every human cell is being defined in terms of its expression of genes and what it does, function, etc. That is leading to an understanding of human biology. And it's, like I said, that it's a perturbation of human biology, of human physiology that eventually leads to precipitation of various kinds of diseases. The information that's being generated in the various Human Cell Atlas projects will eventually enable better disease management. And again, I know at the risk of repetition, I'm going to state that it will save on cost of treatment, it will reduce patient suffering, It'll reduce anxiety of family members because it's no longer across the board treatment, it's more precise, and we hope that it'll increase patient survival. Um, these health benefits needs to accrue across geography, ethnicity, gender, and age. And the Human Cell Atlas is actually trying to promote and ensure that. And that's what we believe is equity in this particular sector of human health. If I just can take one single disease, which is, or one single perturbation of human health, which leads to cancers, the kind of questions in cancer biology that's being addressed by the emerging technology, which is single cell RNA sequencing, essentially starts, we had the understanding was to figure out, what proportion of individuals who are suffering from these various cancers are predominantly because of genetic causes or non-genetic causes, and the interaction between the two. That has been fairly well understood. The various kinds of microenvironmental cues and cell-cell interactions are being understood, and so on and so forth, recurrence of disease, etc. The Human Cell Atlas essentially is trying to understand the cell of origin and developmental pathways in cancer using single cell methods and using single cell biological methods. And we really hope that all of this will eventually lead to better understanding of cancer and therefore better management of cancers. But for this to happen, there should not be any failing on diversity. Genomics has failed on diversity during the past many years in the GWAS studies and the genome-wide association studies. Primarily, that has happened in individuals of European descent. And as we are trying to understand using single-cell technologies, inclusivity is of paramount importance. And that's the reason why the equity working group was established early on in the HCA, and many of us who have been involved with the equity working group are actually trying to promote inclusivity in the working of the Human Cell Atlas. The roadmap of the Human Cell Atlas is that we are looking at various organs of the body, various tissues in organs of the body, and these are not being looked at in one particular group, one particular population group, but it's across ethnicities, genders, ages, and like I said, that we are collecting tissues from various organs, dissociating these tissues into single cells and trying to understand the gene expression, and thereby trying to understand the various kinds of cell types and states in the organs, in the tissues and organs of the human body. That leads to, in order to be able to do that, a lot of experimental and computational methods have had to be developed. These are still being sharpened. There are many new methods that are actually being formulated, both on the bench as also in the computing lab. All of this, of course, single-cell genomics is expensive, remains expensive still, and therefore many funders have been contacted by the leadership of the HCA, and funders have come forward, and some of the equity dimensions and some of the activities that we are able to undertake is because of this understanding of inclusivity by the funders who have actually pitched in money in order to promote equity. HCA is actively promoting equity and is actively taking actions to share knowledge and knowledge of concepts and technologies. And of course, these workshops like this Latin American workshop, this is of course the second workshop. Earlier there was another that was held where these concepts and technologies were discussed. Both concepts and technologies are actually evolving, and therefore it is imperative that that there be an ongoing series of such workshops, and these are being organized by the leadership of the Human Cell Atlas, by the Equity Working Group, and these roadshows are primarily being held in low and middle income countries. These workshops are or the equity activities are meant to empower scientists from underserved and less endowed countries to participate in the activity in HCA, in the science of HCA, And the idea is to engage them as partners in research and translation, provide them with access to these technologies. These technologies are not easy to access. They are expensive now, and they need a lot of training to be able to use these kinds of technologies to the benefit of biology. So providing access to these technologies, educate, build capacity, and be educated. And the be educated part is also important. It's not like a one-way traffic. Those who are participating in these kinds of activities that promote equity are themselves being educated by through these various kinds of workshops and road shows that we're participating in. HCA is also enabling provision of less expensive and more portable technologies and reagents, preferentially to the LMICs. Like I said, that the reagents and the platforms and the reagents are both expensive. Also, there has been a lot of challenge in terms of doing single cell analysis because of lack of portability. But right now, there are newer technologies that have become available that make these kinds of technologies more portable and more usable. The Human Cell Atlas is actually committed to serve humanity. And that commitment comes in the form of a manuscript that we have developed. The equity and ethics and other active members of the organizing committee, etc., have developed a manuscript. The manuscript is available in open access, but we have submitted the manuscript to BMC Genomics, and it's under review right now. The main points that we have made in this Human Cell Atlas, it is about 10 pages long. The manuscript, essentially what we have committed ourselves to is that members of the HCA are not going to do helicopter science. We've had enough of helicopter science where people from certain nations will descend on certain other nations, take the samples out, and that's the kind of helicopter science that has been practiced in the past. But right now, it'll be the focus will be on local needs through local collaboration. So it's not like taking samples out of the country and going elsewhere, analyzing the samples. The main emphasis will be to first understand the local needs and also to empower the local scientists to be able to analyze the samples, to be able to draw inferences, possibly in a collaborative manner, because left to themselves, these are difficult technologies to use, and therefore these are best done in a collaborative mode, but best done locally as well, and that's the way that equity is promoted. The membership of the Human Cell Atlas is open to scientists globally. It's a very open organization, so anybody who wants to become a member of the HCA can easily become by just registering. And membership also allows, first of all, exchange of ideas, exchange of protocols and concepts. So it's a good idea to actually become a member of the Human Cell Atlas if one is interested in biology. The third point that we have made is that we need to embrace, consult, and work under the leadership of local scientists. Even though the HCA has leadership, but when it comes to individual projects in multiple geographical regions, the leadership needs to be provided by the local scientists, and that's how it will improve local science. And that's the whole idea, that the leadership will be provided by the local scientists, and to the extent possible, the samples that are collected, the single cell RNA sequencing work, etc., will be done in local laboratories, and that's the whole idea. So, in order to for the Human Cell Atlas to impact on biology, to understand cell by cell, the human body cell by cell, one needs to disseminate these various kinds of concepts and technologies such that the whole of humanity is served, such that the benefits are not accrued to a portion of humanity, but globally. So that's the focus of this manuscript, which we hope will be accepted and published soon. We also need to understand whether it's one thing to say that this is what we've been doing and we are trying, but are we actually making any progress? So what are there some metrics? Are there some yardsticks by which we can ascertain whether we are making any progress? So we are, as a matter of fact, developing a set of metrics to measure and track our progress over time. Our progress meaning progress of equity in HCA over time. And the kind of metrics that we are thinking of, and we are open to discussion, open to suggestions. The diversity of investigators participating in the Human Cell Atlas is one metric. By diversity, diversity of regions from which these investigators are participating in the HCA. The donors, of course, the HCA needs samples, and the diversity of representation of donors, geographical, ethnic representation of the donors is another metric of promoting equity. The extent of populating with anonymized data of diverse groups in the database, all of the data will go into a database, and they'll be in an anonymized fashion, but there will be, some kind of an identity of the population groups from where individuals have been sampled and their tissues have been analyzed, their cells have been analyzed. So the extent of the diversity of in the database, the diversity of the population groups that are represented is another metric that should improve over time. The diversity should increase over time, and that will be a metric of progress. And also, we are trying and understanding, having promoted HCA in local laboratories through local leadership, etc. Are there some benefits that are accruing to the local participants or to the local scientists because of their participation? Is the science in those regions improving? Understanding of science and understanding of how to analyze data, etc. So the benefits can accrue at two levels. One is that the level of promotion of science, improvement of science. The second is at the level of the society, and both need to be tracked over a period of time in order to figure out whether we are making progress. So the whole idea is that we need to develop a set of metrics such that equity does not is not a matter that's bound to the lips, but actually we are able to track it and see that this is actually being promoted, and it's improving over time. We actually had published our views on equity, the Human Cell Atlas on equity. There were a set of 10 points, 10 lessons that we had learned. And this has been a couple of years ago that we published this in Nature Medicine, and this is available. This is in the open access, so anybody who wishes to understand. And it's the commitment to humanity is a sequel to the lessons that we have learned. And we believe that that commitment, the metrics that we are developing, will actually show that the Human Cell Atlas is serious about promoting equity and is seriously taking steps in order that there is diversification and there is promotion of equity. So I'm going to stop here with these very general terms of what I've said about equity. But my participation or our participation in the Human Cell Atlas has also improved our science through collaboration, through again not necessarily through funding, but primarily through collaboration, understanding of how these things are done, etc. And we have been beneficiaries of this kind of collaboration and knowledge. And I wish to share with you some results of single cell RNA sequencing work that we have done in India, and we have focused primarily on oral cancer. Although the Human Cell Atlas is trying to understand, the human body cell by cell, it's kind of important that we address a specific disease if we are supposed to generate funding locally. It becomes very, very difficult, and I've said this in the HCA organizing committee. It's become very difficult to generate money for for understanding or reconstructing the human body cell by cell unless it's tied to some kind of a disease. So in our work on oral cancer, we are focusing on oral cancer, but also trying to understand heterogeneity in the normal oral cavity and the normal cells in the oral cavity. So in terms of our understanding or our single cell transcriptomic work, we are actually trying to generate data simultaneously on the diseased tissue as also on the normal tissue. That's the only way that we could generate funds locally to be able to participate in the Human Cell Atlas. Of course, right now, after we started this work, we now have collaboration with other institutions on other kinds of projects in the HCA where we don't necessarily need to choose diseases, but that funding has come from international funders, and so we are able to participate just by looking at the normal tissues and the tissues derived from normal individuals without disease. So I will now describe to you some of our work. Initially, the work concentrated on DNA sequencing from bulk cells and DNA sequencing and RNA sequencing, bulk RNA sequencing. Slowly, over a period of time, we have moved over to single cell RNA sequencing, primarily because we were understanding that there is a lot of heterogeneity, cellular heterogeneity within the oral cavity, both in the disease state as also in the normal state. Let me describe the disease to you. Squamous cell carcinoma of the oral cavity is the eighth most common cancer in the world. About 260,000 new cases arise annually. About 2/3 of them arise in the developing countries. It accounts for about 128,000 deaths annually. It's related to tobacco usage. About 1/3 of all tobacco-related cancers in India is accounted for by squamous cell carcinoma of the oral cavity. Although between 20 and 60% of cancers of the oropharynx is associated with human papillomavirus, as I shall mention soon, that in the specific carcinoma that we are looking at in the oral cavity, HPV involvement is zero or very minimal. So in India, compared to the West, the site distribution of oral cancer is very different. In India, primarily the oral cancer happens or affects the gingivobuccal complex, which is really the oral cavity, epithelial region of the oral cavity, the floor of the mouth, etc. And it's excluding it's essentially the entire oral cavity without the tongue and floor of mouth. In the West, it's predominantly the tongue and the floor of mouth that's impacted on. The gingivobuccal complex is only, so it's essentially a reverse scenario between India and the West. And also, if you look at the nature of usage of tobacco, which is very strongly linked with oral cancer, in India, it's predominantly tobacco chewing, while in the West, it's predominantly tobacco smoking. Whether this can account for this reversal in epidemiology or of the region of the oral cavity where there is which is affected, we do not know. But this is also something very interesting, that the nature of usage of tobacco is quite different in India compared to the West. We looked at about a set of 250 patients. Most of them were male. 72. Most of them were middle-aged, between 40 and 55 years. Most of them presented very late. They love their tobacco. They keep chewing until they can chew no more. And the disease by that time, the disease has progressed quite late. And most of the patients who came to us were at stage 4 disease. Most were exposed to tobacco. HPV infection was nearly absent. We did, over a period of time, like I said, our sample size is just a little over 250. We are targeting about 500. The oral cancers, the tumor suppressors are the major drivers of oral cancer, and the genes that are involved in oral cancer. The driver genes are, of course, P53 is at the top. 64% of our oral cancer patients are driven by mutations or alterations in TP53. Then we have a set of patients, a set of about 8 to 10 genes that explain a large, about 10% of recurrence in by mutations in these driver genes, but also there is a long tail of mutations. And given the kind of sample sizes, we are able to detect driver genes in about 3% of with about 3% of recurrence. Most of these genes behave as tumor suppressors. TP53, for example, as all of you may know, that TP53 behaves both as a tumor suppressor and as an oncogene. In the context of oral cancer, it behaves as a tumor suppressor. Um, and the way that we another way of understanding that it behaves as a tumor suppressor is if you map the mutations on the genes, there is clustering of mutations, and as you can see that on TP53 there is clustering of mutations, even though there are mutations that happen throughout the gene, but there is clustering of mutations. In all of these genes, one can test for clustering, and one would find that there is clustering of mutations in these genes, and thereby bolstering our understanding, or bolstering our finding from other kinds of analysis that these driver genes behave as tumor suppressors. So essentially, tumor suppressors drive oral cancer. We also can look at heterogeneity in patient survival, and what one finds is that the patients are actually grouped by primarily three genes that drive these clusters or clustered these patients. One is a P53 cluster, the other is FAT1 with or without mutations in caspase 8, and the other is a heterogeneous group of driver genes or alterations in the heterogeneous driver genes that form the third cluster. If we look at survival of these various branches of each of these clusters, the survival probabilities are quite different, are statistically significantly different, which essentially means that it is important for us to understand what kind of genomic alterations have taken place in these patients, such that one can actually figure out that make a prediction of the survival probability of these patients by knowing the molecular defect that has happened in these patients. So again, we've done some cell line experiments to understand what happens when there are alterations in these various driver genes. The recurrence frequency distribution of driver genes has a long tail, as you can see. There are many mutations in genes that occur in less than 2% of the patients. And this long tail essentially means that there is a lot of heterogeneity in mutational profiles or mutational backgrounds of patients. Incidentally, these are mutations that are driver mutations, that we are not talking about passenger mutations at all. So these are mutations in driver genes that has a long tail. So essentially what it means is that there is a lot of heterogeneity, and this heterogeneity now needs to be dissected even better. And what we realized is that if we take if we look at mutational profiles of each patient, they are quite different. And so what we next want to do is to look at tumors taken from patients and look at the cellular composition of the tumors, and understand the diversity of cell types, and thereby understand how much of heterogeneity is there in the tumors. This heterogeneity, of course, we know leads to variation in tumor growth that impacts on therapy response, metastasis, and probability of recurrence. The heterogeneity also influences interpatient variability in survival and treatment. So it's important that we felt that it was important or a natural consequence of the findings from our bulk DNA and RNA sequencing work was to now move into characterization of cell types and cell states in each of these tumors. And so we naturally moved into single cell RNA sequencing work. So our objectives are to profile cell type diversity in the oral squamous cell carcinoma of the gingivobuccal region, to identify gene expression programs in different cell types, and to investigate cellular interactions within the tumor ecosystem. I won't be able to describe to you in gory detail all of the findings of our work, but I will summarize them in some meaningful way, hopefully. The number of patients that we've been able to look at is about 12. We've taken about 50 to 60,000 cells from these 12 patients, and all of the results that I'm going to describe to you is based on single cell analysis of these about 60,000 cells drawn from these 12 patients. Buccal mucosa, as you can see, is what we are looking at, but in a quarter of the patients, in 3 of the 12 patients, there was a precancerous lesion that we knew led to the formation of the tumor, and this precancerous lesion is called oral submucous fibrosis that preceded the formation of the malignant tumor in 3 of these patients. In the other 9 patients, there was no evidence of a precancerous lesion, or at least no known evidence of a precancerous lesion. The grades were variable, and most of these patients had well-differentiated squamous cell carcinoma, which is the brown region, and followed by moderately differentiated, and then one patient had infiltrating squamous cell carcinoma. So the grades are also quite high. These are all high-grade lesions or tumors, and those are the kinds of tumors that we have analyzed. We've classified the cells. We have drawn cells, and as I said, that roughly 5,000 cells from each individual. We've classified these cells into malignant and non-malignant cell types. The standard method of classification is to use infer C and B that relies on the fact that there are copy number alterations in cells of the in the malignant cells, and therefore use copy number alterations in order to identify malignant cells. We have also devised another computational method, which we have called as the distance profiling method, that looks at the gene expression levels or gene expressions of epithelial cells, which are the ones that lead to become malignant epithelial cells based on the reference non-epithelial cell. And based on these distances, so this is on the x-axis are the distances, on the y-axis are the number of cells at that particular distance. And as you can see that there is a clear antimode, and on the right side of this antimode are the ones that are quite distant from non-malignant, non-epithelial cells, those are the ones that we call as malignant cells. So there are 2 different ways that we have classified cells into malignant and non-malignant. If you look at the profile of of OSCCGB, what one finds is that there are essentially 7 cell types. Of course, the malignant cell type is large, and there are mast cells, B cells, myeloid cells, and so on and so forth. Each cell type has representative cells from all of the patients. So all of the tumors contribute to these various cell types. If we look at malignant cells, these are the malignant cells. If you look at the malignant cells, it's interesting that the malignant cells cluster by patient. It's not like these clusters are extremely tight, but by and large, tumors of patients have characteristic features that make them cluster by the patient type as opposed to the cell type. On the other hand, the non-malignant cells cluster by cell type, and that was an interesting finding. We looked at epithelial to mesenchymal transition in oral squamous cell carcinoma, and we found one of those clusters was enriched with cells that had partial EMT and hypoxia-related genes. As you can see that BIM, If1A, these are hypoxia-related genes and partial EMT-related genes. In this one cluster, in cluster 14, there was a concentration of these kinds of expressions of these kinds of cells. The higher correlation of hypoxia was observed in this particular cluster, cluster number 14, and we identified the genes that might actually play a role in hypoxia-mediated partial EMT in OSCCGB. So partial EMT is an important sort of a driver program in OSCCGB. We looked at these non-malignant cells, and essentially, we found that there was a lot of diversity in myeloid cells, in fibroblasts, and so on and so forth. Don't have the time to really get into the details of this, but each cell type had a lot of heterogeneity. The T cells were abundant in the tumors with diverse cell types and infiltration. The dominant B cell types, even though there was a lot of diversity, some amount of diversity in the B cell types, the dominant B cell types were the plasma and memory B cells. The myeloid cell types also in the previous graph. I didn't spend any time in the previous graph, but there was a lot of heterogeneity in the myeloid cell types also, and in that subtype, M1 macrophages are the most common. And the macrophages in tumors exhibit an intermediate M1 to M2 polarization state. Diverse endothelial cell subtypes known to perform a variety of cellular functions were also present. So essentially, we were able to identify some interesting programs that are interesting changes that take place in OSCGB, and we were able to also do. So that's essentially in the normal tissue, in the normal oral tissue. But in the malignant cells, like I said, that the malignant cells essentially grouped by patient, and there also we identified that there are essentially two groups, as you can see, and interestingly enough, the group one individuals in group one are the individuals who had prior precancer, oral submucous fibrosis that progressed to oral cancer. So essentially, those individuals, those patients who had a prior pre-malignant lesion and progress to oral cancer, they have their single cell profiles are quite different compared to those that did not show any precancerous lesion in the oral cavity. We subclassified the malignant cells, and essentially fetal and germline epithelial cells were abundant, and malignant cells are abundantly expressed in genes of the immune related pathways. Immune-related pathway genes are abundantly expressed in the malignant cells, which is actually good news for immunotherapy. To summarize, gingival oral cancer, which is the predominant cancer among males in Southeast Asia, exhibits large intratumor cell type heterogeneity. Those tumors that are derived from precancerous lesion of the precancerous lesion, which is the oral submucous fibrosis, they exhibit a distinct expression profile compared to those that are not associated with this precancer. So what we are now trying to do is take a number of oral submucous fibrosis patients and find out which of the genes that are expressed early on, whether or not we can prevent precipitation of frank malignancy by managing those patients that show expression levels of some of these genes that we have found in this group 1 set of cells. Fetal cellular reprogramming and partial EMT play major roles in oral squamous cell carcinoma of the gingival region, and in the process of tumorigenesis, fetal cellular reprogramming and partial epithelial to mesenchymal transformation play a major role. So that's essentially what I really had to present to you. I'm very glad to have participated in early on in the Human Cell Atlas, without which it would be almost impossible for us to generate this kind of data and to understand oral cancer in this kind of depth, primarily because we needed people who actually were conversant with the technologies, who were sort of inventors of these technologies, to handhold us, to take us through the various steps. And this could not have been possible without promotion of collaboration and equity that are considered of paramount importance in the Human Cell Atlas. So I really hope that this particular workshop will help scientists in various countries of Latin America to be able to participate in single cell activity and to be able to understand diseases of local interest at the single cell level, while simultaneously also understanding what happens to the normal body that gets cells in the normal body that gets perturbed in order to precipitate a disease state. Again, I thank the organizers for giving me a chance to to present my work, and most importantly, to present the fact that equity is of paramount importance in the Human Cell Atlas. Thank you very much.